This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 146, recorded on August 26th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Joining me today from North Central Florida is Rich Condit.
Howdy there, Vincent. How are you?
I'm well. I missed you guys.
Well, you were having a good time. We missed you too.
I had a great time, but I listened to your TWiV 145. It was great.
Well, it came out okay, I thought, but it's certainly not the same without you.
Yeah, but I think, in fact, Alan talked more without me there. Maybe the fact that he's a former student of mine plays into that.
I think there was space to fill. I think we were both sort of pumped for it.
Yeah, well, over on TWiV, they did it without me twice, and I also found that they talked more without me there. So maybe I'm inhibitory. I'm a negative regulator. Who knows?
You're a good part of the whole deal.
Well, I'm glad to be back.
Make it happen.
I enjoy it. I miss doing it. I did have a great time in Europe, but glad to be back. Now, Alan is off on vacation, right?
Yeah. Is that well? He's not here, right? I was wondering where he was. He's on vacation?
I think so. Yeah, he said he was going to be at the Cape this week.
Okay.
And of course, he's going to get a hurricane.
Yeah, that's not good.
Let's bring in our special guest. Here, we always talk and leave our guests in the background for a while. Our special guest is from the University of Oklahoma, Abbie Smith. Welcome to TWiV.
Hi.
Hey.
Yeah, I'm the one from Oklahoma. I'm the one that's supposed to be saying howdy.
Howdy. Are you from that area originally?
I'm from Central Missouri. So the Midwest, it's all kind of the same.
Yeah, Rich is a howdy kind of guy, right?
Yeah, I don't know where that came from. But there it is.
Well, Abbie, you're also known as ERV.
Yes.
Online. You've got quite an online presence. You have a blog over at scienceblogs.com/ERV.
Yeah.
And you're a grad student, right?
Yes.
What year
For
are you
a long time. This is my 5th year. So hopefully I'm going to be graduating sometime next spring. But the second I say that, I will be here for another 5 years. So it's penciled in on the calendar.
And you're a grad student in, what is it, Department of Microbiology?
Microimmuno.
Microimmuno, just like my department here. And what do you work on?
I study the evolution of HIV and how we can learn more about the evolution of HIV and apply that to vaccine design.
So who's your advisor?
John West.
John West, okay.
Yeah. Not the one at the Discovery Institute. There is another one there, which is kind of funny in the lab sometimes, but definitely not him.
Okay.
So it would actually be good if we can get to this second paper today because Abbie might have some insights into that.
I'm sure.
Oh, yeah, definitely.
All right, we'll make a point to get to it, so I'll stop talking, Rich.
Nah, it's okay. It's part of the... So I want to know about how Abbie got into blogging. How long have you been doing this?
I started blogging when I moved to Oklahoma in 2006.
Okay.
I just moved down here. I didn't really know anyone. And all of these things were happening around me regarding creationism and that sort of thing. And so I kind of needed a place to vent.
Okay.
And so I turned to blogging. And, then I also kind of turned it into an opportunity to educate the public about viruses and things they were hearing in the news about viruses and some of the cool things that were happening in labs all around the world that aren't getting covered by the news that I think the public would be really excited about. So it's kind of turned more into that.
So do you have a history writing? I mean, is writing something you'd have always liked to do or something like that? Because, if I was looking for a place to vent, I wouldn't necessarily start a blog, right?
Well, it's almost like screaming into the void. You know, on a blog, you can just go and get it out. And I am definitely not a writer, and I don't necessarily characterize myself as a writer.
Okay.
There are definitely some science writers out there that are just excellent and just fun to read. I'm definitely a blogger as opposed to a writer.
Yeah, it's an interesting distinction, Rich. Many people use blogging as a venting platform and don't necessarily care about the writing. And it works pretty well as that, you know, and then others use it to write. But you said, Rich, if you were going to vent, you wouldn't write a blog. What would you do?
I'm not sure. I'd probably just sit there.
No, you'd do something.
You think so?
Well, blogging is easy. That's
I'll
the
think
thing.
about it.
I mean, now there's no energy level to get over because it's so easy to do.
Right.
I started mine in 2004 for much the same reason. I wanted to teach people. I figured I'd written some books and I wanted to spread the word.
Blogs are so fast and easy and current. So something comes out in the news and I can cover it immediately. You don't need to wait for a blog or for, you know, a magazine to get published or a book to get published.
You have a pretty good audience too, because you get a lot of comments.
Well, I was going to ask about that. So how much feedback you get to your venting?
Well, not all of it's venting. A lot of it's education too.
Right.
And commenters are great about asking questions. I almost wish that people would be more vocal about asking questions, but really the biggest wall I have come up against is that people think their question is stupid, even though there are probably hundreds of other people out there wondering the exact same thing. They're too afraid to put it out there.
That's what I always tell students in class, you know. I mean, you try and tell them there's no such thing as a stupid question, but nobody listens to that. But sometimes they'll listen when you tell them, you know, if you've got this question, chances are half of the people in the room have the same question. So do us a favor and ask the question.
Exactly. And I mean, you know, I deal with creationists very often online and in real life. And sometimes they genuinely do ask stupid questions, but the ones that I get from science blogs readers who are interested in science and really have a very fantastic layman's background in science, they always have the most fantastic questions. And I wish more of them would speak up.
Yeah, you get only a few percent or 1% of your readers actually participating. So there are a lot of them out there reading, but they don't participate at all. How do you balance doing your research with blogging? Because you do it pretty frequently, right?
Yeah, it must take a lot of time.
Oh, it doesn't take as much time as you think, because a lot of times I'll write about papers that I have to read for work or class anyway. And so that just makes it a really good opportunity to educate everyone on that particular paper. But my rule is I don't do any blogging at work. Like work is work time. And then when I get home, you know, maybe instead of watching something stupid on TV, I'm like, well, you know, let's go online and write a blog post about something interesting.
It sounds really level-headed.
Yeah.
I like that.
Almost too much.
So we have you wasting time in the middle of the day here. I'm sorry.
Yes, but no, it's okay because so the way my experiments work normally, you know, I set up an experiment and then I set up another experiment and I set up another experiment, and then by the time the second one's done, the first one's almost ready, and like you kind of have to stagger all of your experiments. But right now I've kind of got this empty space. So I was like, I time my day just perfect. So I'm not really missing anything. But the second we're done, I have to run back in and do more work.
So you sound pretty organized. Is that how you would characterize yourself?
You can't see the desk I am sitting at right now.
It's covered with junk, right?
Yes.
Okay.
And piles, really more piles. It's vertical. But as far as my time goes, you know, to get things done, you really do have to be organized with your time to get things staggered just right so you can get the most out of your day. And
I love that to me is a challenge. It's I get a sort of obsessive compulsive about it. I like trying to cram as much as I can into a day and be busy all the time.
Yeah. But then inevitably something goes wrong with an experiment, or a machine you're wanting to use has a problem with it and you have to spend an hour tinkering with something. So it's good to have your schedule, but it's also good not to be so fixated on it that just everything goes wrong if one thing goes wrong.
So what are you going to do when you grow up?
Well, I want to do this forever. This is
Okay.
fun.
You
I
want to
love
be
research.
you don't want to be a graduate student forever, right?
If they'd let me, it's really not that bad of a gig. I mean, you've got to, especially in this economy. I mean, you get a paycheck, and the cost of living in Oklahoma is pretty low. So it's fun. It's fun. But
So
no,
academic science?
possibly, yeah, or private, or if I can get some Craig Venture money and be Willy Wonka of viruses, that would be fantastic too.
Okay, so research.
Yeah.
And are you going to keep blogging when you do this forever?
Well, I hope so, but things always go out of fashion.
Mm-hmm.
I mean, things change on the internet overnight. So at some point, that might not be the cool thing or the most productive thing to do anymore. But as long as it is, I'd like to keep doing it.
Yeah, I can imagine it's always going to be a good way to teach people, though, right?
But there might be better ways.
Like what?
I have no idea.
How about podcasting?
Yeah, definitely. Well, because people can listen to this, when they're driving to work or like commuting on the bus. That's fantastic.
Yeah, so I'm amazed at how many people have latched onto our podcast, and they tell me, we do a lot of work where we can listen and can't read. So for those people, it's really good if we're commuting, as you say. And, it's different. A podcast like this, we try and make it a conversation. So it's kind of fun to learn that way. A lot of people tell us that. But who knows, you might do that as well. One thing I wanted to touch with you on is that there are not, something we discussed a little bit in our correspondence, there are not that many virology blogs, right?
I know. I don't know what's up with that.
I mean, pure virus like you do and I do. I found two others, one by a professor down in South Africa, Ed Ribicki. You probably know him, Rich, don't you?
Yes.
He does.
We've had this conversation. First time I said I didn't know him, and then
he wrote to us. Yeah,
he wrote to us and said, "Yes, you do." Okay.
Yeah. So he has a viral bloggy, and then there's Rule of Six, which
I just started reading that a little while ago because of Google Plus.
Yeah,
and it's good.
Yeah, definitely.
It's very good.
Very good.
I saw him just before ASV, and he was when I was live tweeting the meetings at ASV. We had a little conversation. You didn't go to ASV, did you, Abbie?
No, us HIV people are.
No, you
are
have your
different.
own meetings, right?
Yeah.
It's a really good meeting. It's really good for connecting, and you should try and make it sometime because those HIV retrovirus meetings are great, but you just don't see all of virology, which is really good, right? And ASV.
Well, the thing is, I do have lots of outside interests. Like, I love learning about all viruses. I love viruses. And that's one thing that I actually do get in trouble with at work because my boss really wants me to focus on my field and what I'm doing. And then he walks in and I'm reading a rabies paper and he's like, come on. It's like, focus.
Yeah, I don't get in trouble because I don't have a boss, so that's good. I can do all this stuff. Someone told me once... she walked into her cell culture room and everybody had their, iPods on. And she said, "What are you listening to music for?" And they said, "We're not. We're listening to TWiV."
Nice.
So that was pretty cool. I don't know any other virus blogs. I mean, there are mixed ones. There are microbiologists who do viruses, but no pure. But if we're wrong, if anyone's listening, another virology blog, tell us.
I post, shout outs for, if you have another virology blog that either you write or you read, it's like, I definitely want to know about it. Yeah. And I just don't hear about them. So definitely if someone out there has a blog, a virology blog, or knows of one they really like, I'd love to hear about it.
Now here, I don't know many students who have blogs at all of any kind. And so what do you think about that in general? Not just virology blogs, but do graduate students blog about what they're working on, or is that pretty rare also?
I don't know. I think it was definitely good for me the first few years of graduate school because when you turn something you've just learned in class or a paper you've just read into a blog post that you're trying to explain what you've just learned to someone else so they can learn, it really helped me assimilate the information, I think, very well.
That makes all kinds of sense. It really fixes it in your mind.
Yeah. So I would definitely encourage it. But at the same time, like we were saying before about balancing your time, some kids just don't have the time for it or, you have to. I always emphasize don't do this at work. It's like this is a hobby. This is something I do at home. but then if you're studying all the time, you don't really have time for that sort of thing.
Yeah.
But if you do, or you're looking for something just a little different, I would encourage other students to think about doing something like this.
I think, as you said, it's a great way. If you're reading a paper, it's a great way to summarize your thoughts and put it on paper, and then you might as well publish it and let other people see it as well. It's not that much extra work.
No. And sometimes you have questions, and then you get readers that have other questions, and it's more of an interactive sort of way of learning.
Yeah. I think blogging has a bad reputation in a way of just something where you vent and tell about what you think of something, but as for teaching, it may be not as recognized that it's as effective, but it is. And one of the things I don't like is that, after a few posts are written, they disappear off the front page, and then no one ever sees them again.
Mm-hmm.
And
you go back and read old posts, and you're like, wow, that was really good.
Yeah, there are plenty of older posts that are good, and I'm trying to figure out a way to make them more accessible, to make it more of a resource than something that's a daily thing that's gone after a day, and then you don't have to deal with it anymore. So if we could figure that out, I think it would be even more powerful a platform than it is. It's the same problem with Google Plus, because you write something and then in a day it's gone and nobody goes back and looks at it. So there is something to be said there. Well, let's go ahead and do a typical TWiV with you and discuss a couple of papers.
Well, sure.
And the first one is one that a number of people have emailed us about, and I know you had a number of requests as well to discuss this paper that came out of MIT not too long ago called Broad Spectrum Antiviral Therapeutics, published in PLOS One. That is quite a title.
Yes.
Actually.
Yes.
The first author is Todd Ryder, and it's Christina Zook, Tara Butcher, Scott Wick, Jennifer Pancoast, and Benjamin Zussman. I don't know any of these authors. I suspect they are not virologists.
It's from some sort of, it's from the Lincoln Laboratory. It has something to do with nanotechnology or something like that.
Rich, when I was at MIT, the Lincoln Lab had a bad reputation. I don't know if you remember that.
No.
Because they were a biodefense lab.
Uh-oh.
And people didn't, this was in the 70s, 80s, and people didn't like that.
Uh-huh.
So maybe it's changed since then. Got a lot of money from the Defense Department of the U.S. But so I got letters from Jonathan, Dave, Jason, Michael, Alfred, Alex, and Reynard, also Josh. I'm going to read a couple of these.
Connor brought it to our attention, and I got a couple of emails myself. So this is right up on everybody's radar.
Yeah. And let me read a couple of these because there are actually some questions that we should hit on. Jonathan wrote, so he was praising this podcast, and he said, after listening to the first lecture, I found a PLOS One article that states that a research team has drafted a double-stranded RNA that, to the best of my understanding, binds to a viral RNA particle prior to being incorporated into the host's genetic material and then exploits an apoptosis pathway. So that's a little bit wrong, Jonathan, so we'll fix that for you. And he says, any comments about survival of the fittest with respect to virus in light of this? So that's a really interesting question. Dave actually was at ASV and enjoyed our podcast there. Looking forward for another one. He also mentioned the PLOS One paper. Do you think this approach has the potential to be a broad-spectrum antiviral, ignoring for now any possible issues with drug delivery? That's a big one.
Yeah, that's a big one.
And he mentions that he uses TWiV in his course. He's a professor at Dickinson College in Pennsylvania. He uses it to give them background on papers that they're going to read. We also got one from Jason, who's at Personal Genomes, which we know because we've talked about that on TWiV. I got one from Michael. He sent a quote from Times of India. Scientists are developing a new drug which is showing great potential to revolutionize the world of medicine with its ability to cure everything from colds to HIV, although no retrovirus was studied in this paper. That's interesting, right? What else do we see here? Here's one. Alex is an electrical engineer who says we provide a new world that he had no familiarity with before. He said, "Can you comment on this?" And okay, so those are all our reader comments. So this is out of MIT. I was reading, I don't know what you guys think, but just reading the introduction kind of left me wondering if these individuals have ever thought about viruses before. First, the statement, currently there are relatively few antiviral therapeutics. Few to me is a couple, right?
Yeah.
I think there are over 30. I mean, there are a lot of HIV antivirals, right, Abbie?
Definitely.
And there are some for herpes and influenza. So relatively few may be a mischaracterization. Then the first sentence of the paper, a serious threat is posed by viral pathogens.
Yeah.
I would never write that as the first sentence of a paper, and I think either of you wouldn't either.
No.
And do you know what a clinical virus is, guys?
By this definition, I suppose something that you get sick and go to the doctor with.
Yeah.
But I don't see how that's different than
Any other.
something
So
else.
I don't mean to pick on it, but the point is I don't believe these are virologists. Anyway, the general idea here is they made a new kind of protein which is modular. They have one part is a double-stranded RNA binding domain, and there are a bunch of proteins in the cell that will bind double-stranded RNA. And then there is a domain that will activate the apoptotic pathway, that is the programmed cell death pathway. And then finally there's a domain that will help get this protein into cells. So three different domains, I guess, right?
Right.
And
they call it, the approach they call double-stranded RNA activated caspase oligomer, or Draco. Would you pronounce that Draco?
I guess Malfoy would.
Yeah.
With everything, you've got to have a catchy acronym for it.
Right. I'm no good at catchy acronyms. I really.
Yeah, they could. I guess they kind of modified this to be Draco.
Sure.
And that's what everyone's calling it. So double-stranded RNA is generally believed to be found in most virus-infected cells, right? How about retroviruses, Abbie? Do we know?
No. Well, I mean, we know, I think, and I would say no.
Okay.
So the HIV genome is diploid.
Right.
So it's 2 copies of a single strand. And we do know that the structure of that RNA is very important, but RNA structure and double-stranded RNA are different things because, of course, our own RNA, when it's being translated or used for other purposes, has structure. Double-stranded RNA, I think that would be targeted by a pattern recognition receptor, is a little bit more beefy.
Mm-hmm.
And in that sense, I would only think of specifically double-stranded RNA viruses, or either positive or negative since single-stranded, which, of course, in their life cycles would have double-stranded RNA when they're duplicating their genomes or making more messages. So definitely an RNA virus, I would say this might work on, but I would need to see some kind of scientific evidence that it would work the same way with a retrovirus.
Mm-hmm. Yeah, and interestingly, they didn't do that in this paper.
Yes.
Which suggests to me it just didn't work.
Yes, because if I had something like this, the very first thing I would do is test it on HIV, even if I weren't an HIV researcher.
Right. I mean, they tested so many other viruses. They must have tested some retros. Rich, what about pox viruses? Do you see?
Pox viruses make some double-stranded RNA, in particular, late during infection, because there are converging transcriptional promoters and a funky transcription termination mechanism that results in read-through of downstream genes. So converging promoters generate overlapping transcription. You get double-stranded RNA out of that.
Same with adenoviruses, right?
Right, and one of these double-stranded RNA binding domains that they toyed with is, in fact, a pox virus immunomodulatory protein called E3.
Mm-hmm.
The pox virus, we don't have a lot of fancy names in pox viruses. E3, okay? And it specifically binds and sequesters double-stranded RNA, okay? So, presumably the virus knows that knows in quotes that it's in trouble with its own double-stranded RNA, so it makes this protein to bind it up and keep it out of sight of the cellular responses. So yes, pox viruses make double-stranded RNAs, and I gather that there's a little double-stranded RNA made during an adenovirus infection. I don't know the details of that, but based on the transcriptional program of adenovirus, where both strands are transcribed at various times during infection, I can certainly see where there's the potential.
Yeah. Well, of course, this
RNA.
protein PKR, protein kinase RNA activated, double-stranded RNA activated, is in fact activated in adenovirus-infected cells by double-stranded RNAs.
Okay.
And then the virus then inactivates it by making a very short double-stranded RNA called VA-RNA. And this is short, so it only binds one PKR protein. And that is the way you prevent it from being activated, because you need two or more PKR to bind a double-stranded RNA to get it activated. And that's the key to their specificity here. And they make this statement: uninfected mammalian cells generally do not produce long double-stranded RNA greater than 21 or 23 base pairs. And that way, you're not going to get multiple PKR or any other RNA binding proteins to bind. Right. I just wonder if we just haven't found them yet.
Well, interesting question. I don't know.
Yeah, so that's one of the key issues I have with this. So that's the idea here. You make a modular protein. It will bind double-stranded RNA. So if you have an infected cell, you make long double-stranded RNA, and you can have multiple of your modular proteins binding this double-stranded RNA. That activates apoptosis, which kills the cell, and it prevents virus infection.
So key to this, as I understand it, is that the apoptosis-inducing functions need to at least dimerize, if not oligomerize, to have activity.
Right.
And it is the binding of these modular proteins side by side on double-stranded RNA that brings the domains of the anti-apoptosis portions in proximity so that they can dimerize and then do their job.
Right. And then the other issue is getting this protein into a cell.
Right.
Which in general it doesn't happen. Big proteins don't get into cells.
And I think that's a key issue with this whole thing. And I'm not familiar with these delivery methods that they talk about, but apparently they're known things. What are these domains that they use? I'm looking for them here.
I know one is the herpes VP16 protein, which you can add to cells, and it just gets in magically. And then there are a couple of others here. Where are they? For delivery, they're fused to proven transduction tags. So HIV TAT, right, Abbie? TAT by itself can get into cells?
I don't know about that.
Yeah, that's what they write here. The polyarginine can do it. And they don't actually use the herpes protein, but there is a herpes protein that can do the same thing, which is actually amazing. It brings a whole protein in with it. And that is obviously key for this, because they want to treat infections with this big protein, relatively big protein.
Right.
But at the same time, really any cell would take up this protein. And so if you inject it IV, you're going to get it in, vein epithelia. Mm-hmm. And then you're going to get it in the liver, and you're going to get it in kidneys. So, really, I'd be more interested in this in a mouse model, seeing, if you inject it IV, does it get into the brain at all? Because there was just a little girl a couple weeks ago who died from Eastern equine encephalitis. And, knowing whether you need to treat someone like this, if you were going to use this drug, can you just inject it IV, or do we need to get into the cerebral fluid for this to work? How do you deliver it where it needs to be?
Yeah, I would imagine the blood-brain barrier would be a big complication for this. First, for when you needed delivery to the brain.
Right.
And to the right cells,
Right. Yeah, so you make a good point. It's at this point, at least, it's essentially untargeted.
Yes.
Right.
Okay. And so the specificity relies entirely on the fact that double-stranded RNA is basically unique to the virus-infected cells.
Well, in all
Yeah.
fairness, of course, most antivirals can get into many cells,
Yeah, right.
and then they have
They're
some
systemic.
so they have some specificity based on where the virus is.
Right.
But the ones that are licensed, of course, don't have side effects or have minimal side effects, and we just don't know
Don't know about
this one yet. So they make a bunch of these proteins with different kinds of the three domains that we've talked about that can express them. I guess they express them in E. coli, and then they add them to cells. They show that they can get into cells. They can last for a while in cells, a couple of
And they
days.
don't last forever. I mean, that's a good thing as well, because if you're actually going to use this as a therapy, you don't necessarily want these guys sticking around forever. So if you can deliver them where they're needed and then they go away when the infection is resolved, I mean, that's a good thing to know.
Yeah, I mean, I would not want to have an apoptosis-inducing protein around forever. I just think that there are double-stranded RNAs in uninfected cells that can activate these Dracos. We haven't seen it in this paper, but
Well, the thing is, the good thing about what they've done conceptually is that they've taken, the standard pattern recognition receptor and kind of cut out all the middlemen, all of the intermediate messengers that need to go through to get to the apoptotic pathway, which is good because viruses, of course, have evolved with all of this for millions of years. So they've evolved ways of interfering with that pathway. Mm-hmm. And so by cutting out the middleman, you cut out the virus's ability to get around it. So conceptually, that's good. And I forgot where I was going.
it's okay.
That's okay.
No problem. It's a good point. But it is a clever approach. I like the idea of taking advantage of this double-stranded RNA binding activity and making a new kind of protein. I think it's cool.
You got it back, Abbie?
No, I almost had it.
You almost had it. Okay.
I know I had a good point.
It'll come back.
It'll come back.
We should mention that this double-stranded RNA binding activity is nonspecific. It's not sequence-specific.
Right.
It'll bind any double-stranded RNA. So they show when this Draco gets into cells that it induces apoptosis. All right, and they measure apoptosis. Now I forgot how they measured it. I have to go. Anybody remember offhand?
I did not actually look at exactly how.
All right, it's a standard apoptosis assay. Here you go. They're looking for caspase glow. They're looking for caspase activation using a commercial kit. Okay, so that's how they measure apoptosis. So these things induce apoptosis. In fact, that's figure.
Figure 3. They chuck in double-stranded RNA.
Yep.
And along with the protein, and show that if there's both present,
it induces apoptosis. Right.
Apoptosis.
And they start looking at viruses. They look at one of my favorites, rhinovirus, and they show that if you add this to rhinovirus-infected cells, you can inhibit the decrease in cell viability, which the virus causes. So the virus usually kills cells, but these Dracos will block that. It looks pretty good to me.
Right.
And they can show you can treat 6 days before, 4 hours after, 1 day after, and 3 days after infection.
That's pretty extraordinary. Of course, what they're doing here, as I recall, is they're throwing, in those particular experiments, they're throwing a fairly low multiplicity. They're throwing, a few hundred PFU onto a well with a lot of cells.
Yep.
Okay.
Yep.
So you're looking at multiple cycles of virus replication. So it's not as if you've infected all the cells and wait for 6 days and add the drug and it stops the virus. No, it's more like, you have a fair number of uninfected cells, so you're probably stopping the spread.
Yeah, I think that's important because if you did this at a high MOI, I'll bet the effect would be much less pronounced. And they should have, I think, done that to support the idea that this is what's happening. They have a model which says, what we think happens is in these virus-infected cells, we add Draco, it kills them, and that prevents virus spread. But they don't actually do any experiments to prove that. Mm-hmm. You also notice that if you treat after infection, it's not as effective.
That makes sense to me.
Yeah.
You know, I would think that when the virus gets a head start, this is going to be a race, right?
Yeah.
Well, remember,
the
though,
virus-
if this is in people, you're rarely going to treat them before infection.
Well, I was thinking about this the whole time I was reading this paper, because we had this discussion a while ago. Usually by the time people show up in the clinic, they're showing up because of the immune response, because so many of the symptoms that you see are the immune response, which means that basically the virus infection has already peaked,
Yeah.
and you're coming down from it. So with really acute infections like rhinovirus infections or influenza virus infections. So in order to be effective, you either have to have some way to, I don't know, diagnose a virus infection very, very early and have something really quick, or have something that'll work late after virus infection, which is less probable.
Yeah. Well, in fact,
Well, what about,
go ahead.
sorry, as a prophylactic, I mean, these days we're dealing with all of these individuals who are being exposed to measles and that sort of thing when, infected individuals are coming back from overseas. So maybe, the CDC could say, hey, if you were on this airplane, you should go to your physician and get a shot of this stuff to see if you can prevent them from getting ill.
Sure.
Yeah, you can imagine those kinds of scenarios. But I think probably the vast majority would be acute infectious situations where, like
Definitely.
flu, But it's tough because of the diagnosis problem. In fact, all the viruses they look at are acute viruses. And they mentioned this may be good for chronic infections, but they didn't test any of them. And maybe that would be like Hep C, long-term Hep C infection. Maybe this would be good for that.
Yeah.
I guess hepatitis B doesn't make double-stranded RNA, does it? I think it just transcribes one strand.
Mm-hmm.
Yeah.
So
yeah, might not work.
Okay.
They didn't test that either, but of course that's harder to do. Hep B, at least, no cell culture
So I
system.
think everything that they've done here are RNA viruses, except they did a mouse adenovirus.
That's right.
And it was effective against the mouse adenovirus as well.
Let's see, adenovirus. They did a human and a mouse adeno, yeah. So they went on and did a lot of other viruses. Rhinos.
Oh, they've got a table
here.
Right?
They did dengue, flu, a variety of interesting arena and bunya viruses.
Right. So yeah, the only DNA viruses that they report on is an adenovirus, but that makes sense.
Right.
Okay.
All right. So another experiment they did is, do these things do anything to cells? Right?
Mm-hmm.
Are they toxic? And they say they're not toxic, but they just do a cell viability assay, which depends on the cells being metabolically active. It's a typical kind of assay kit that you can buy to do this. And I'm not sure that will really be sensitive enough to tell you if anything is going on.
Well, and these were cell lines as well, right?
Yeah, they
Right.
are. Obviously, you want to do it in animals. So they do have one animal experiment. They infect mice with influenza virus.
Actually, they got two. They do one experiment with intraperitoneal
Right.
administration of the drug, and another with intranasal.
Mm-hmm. Okay, so this strain PR8 is basically a mouse-adapted strain. It kills mice, however you put it in. And, they show that you put Draco in mice intraperitoneally, and it gets to liver, which is not surprising, right?
Yeah.
Everything goes there. Kidney and lung, and they didn't look anywhere else. So your question, Abbie, about the brain is a good one.
Yeah. Well, I mean, I'm thinking of all the places where we need drugs.
Yeah.
And anytime a virus gets into the brain, that's definitely a place where we could use some help. So that would have definitely been one of the places I looked.
Right. And they might have, and they perhaps didn't find it because they don't mention it.
But then I
at
want to
all.
know that. I want to know that. And they should want to report that as well. If they, I would have to imagine that they didn't. Because even if it were negative, I mean, that doesn't mean that you couldn't administer it some other way and it would still be as effective.
Right. So they treat the mice and then infect them with PR8, and it has an effect on survival. The numbers of mice aren't huge, but the results are pretty good. And
they got 12 or 13 mice in a group, and
Yeah.
they've got they're killing 90% of the mice with no treatment, and they've got 60 or 75% survivors with a couple of different of these Dracos.
Right.
That's pretty good, and that's with intraperitoneal injection. And I'm wondering, I've got to look again and see what order did they add virus and then.
So they have -1 through day -1 through day 3 is the treatment.
Okay, fine.
So 1 day before, and then through day 3 after infection. Right. And it works pretty well. And they also look at virus titers. There's a good reduction in virus titers as well. And they have that written in on these graphs here. So buffer treatment, 12,000 PFU per mil. That's his lung titers. And then the different Dracos bring it down to 1,200 or 340 PFU per mil. So there's clearly an effect, is the bottom line. And they say the mice were okay. They
Right.
say there's no problems, but it's not even clear what they looked at.
Yeah, I mean, I would want to see some lung biopsies or something like that just for comparison.
Yeah, I mean, I know people at farm companies who do toxicity, and this is just, this isn't anything.
Yeah.
They really have to do a lot. I'll bet there is some toxicity. That's my bet here, that there is some toxicity or some side effect that's going to prevent this from being used in the long term. I don't know, but that's just my feeling, because I think that this cannot be specific for virus-infected cells.
So then they also do an experiment where they do the administration of the drug intranasally.
Yeah.
And they assay for drug in the lungs at different times afterwards in different concentrations, or different Dracos. And they do the same thing with surviving mice. The experiment is set up so that they're only getting, with their buffer control, that is no drug, only getting about 50% mortality. It's interesting that experiment only goes out 10 days, and the mice are still, according to the curve, dropping. I'm wondering why that experiment didn't go longer.
Mm-hmm.
And they've got better survival with the Dracos. It's not as impressive an experiment as the intraperitoneal, but they're getting an effect, apparently. So it's pretty interesting.
Yeah, I'm just looking at this. They say day -1 through day 3. I guess they give it to the mice at every day, -1 through day 3. I see.
Right.
So again, that's I don't know if that -1 is important or not, because again, you're not going to be treating people with few exceptions, as we talked about, at day -1. So I'd like to see at the time of infection only, or a day after infection, 2 days after infection only. That sort of thing.
Or when symptoms start to appear. I mean, something that would be more clinically relevant.
Right, exactly.
But this all has to come under the heading of proof of principle. There's obviously a lot left to do. And I'm astonished that it works at all, actually. When I read sort of the lead-in, I thought, whoa, like, this can't work. And so I'm surprised, in particular, the mouse experiments, that they're getting any effect at all. So it's pretty interesting.
Yeah, it's a neat concept.
Definitely. And in the paper, I mean, normally when I see headlines that came along with this paper and, Yahoo Health or whatever, I'm anticipating reading a paper that is only in tissue culture. It's not even with any primary cells. It's only with the cell line. And there's a lot of hype with not a lot of data.
Yeah.
Right.
And I will say with this paper, conceptually it's definitely interesting. And they do have, the tissue culture, but, at least they had an animal model where they tried to get some preliminary data. And I think it could be cool in the long run. I mean, if this works, it'd be great. But it's still definitely preliminary. Right. And there's a long way to go before this would actually be something that a patient would get in a clinic.
I'm always amazed that people can come up with this kind of stuff. This is another one of
Oh
those
yeah,
things where
yeah.
if a student came up to me and says, "I want to do this," I would say, go. What are you smoking? Okay, go,
Mm.
go back to work. Label your little tubes, run your gels. Don't bother me with this stuff.
Yeah.
Okay, and I'm amazed that it works at all. It's pretty interesting.
Yeah. I think the hype in the press was a bit much, but
Hmm.
that's probably partly MIT's fault.
Yeah, absolutely. I mean, one
Right.
of the biggest hypes comes from MIT itself.
Yeah, they put out a press release, and then broad spectrum antiviral. That's always a big one.
Hmm.
Yeah, I'm totally used to that. With HIV, we're curing AIDS, or we've got an HIV vaccine around the corner literally every other day.
Yeah.
So this was pretty standard press release for me. So I was excited to at least see a novel idea and more data, really, than I was anticipating.
Right. So going back to Jonathan's email, then I think now you understand, Jonathan, what's being done here, and you can look at your email and
Yeah, one of the I gather Jonathan is an amateur at this and has been listening to your lectures or something like that.
That's right.
So RNA viruses generally, with the exception of HIV, don't get incorporated into the host genetic material. Okay, so that's a little mistake there that you can.
Right. And the double-stranded RNA is produced during infection.
Right.
It's not in the particle.
Right.
Right. It's
Exactly.
produced
Except in
infection.
reovirus,
Yeah.
it's in the particle. But Rio has a good way of sequestering that from the cell by basically keeping it in the particle all the time.
Yep.
Those are subtleties. But this, it's a common mistake to think that virus genomes all wind up getting incorporated into the host genetic material, and that's actually a rarity. So that's important to understand.
An interesting question here is, can you imagine a virus evolving to be resistant to this?
Right. That's Carla brought that up in her little quote in the Times article, and I think that's one of the strengths of this is that you're tinkering with a cellular pathway, basically, and so it's harder to imagine how a virus might come up with resistance. Though, I don't know. Viruses.
So they do. They always do.
Right. They have a lot of immunomodulatory tricks. Okay, so they may find ways to deal with this, but this is pretty radical. It's not like you're just tweaking a little pathway or something like that. This is a real novel change in the intracellular milieu. Okay, so the virus would have to come up with a pretty drastic change to be resistant to this. It's hard for me to imagine how it would be done.
because you can't just change the sequence of your double-stranded RNA because it will still bind the double-stranded RNA binding protein.
Right, and it's not like they're just tweaking as Abbie pointed out earlier. It's not like they're just tweaking a pathway that the virus may already be clued into, so the virus can just make an adjustment to get around that. Okay,
Right.
It's a completely novel mechanism that they're introducing.
But Rich, for some viruses do encode inhibitors of apoptosis.
Absolutely. So you can imagine that they might be able to modify those in a fashion to get around this.
Whereas those viruses that do not are not likely to acquire such an inhibitor of apoptosis, right?
Right.
And I would imagine that let's say this does actually make it to the clinic. The drug would probably be reserved for an elderly individual or maybe an infant that can't get over influenza or something like that. Whereas to develop resistance to a drug like that, it would need to be constant. So I'm thinking more of if everybody took this drug all the time, then you would really have selective pressure on the virus population to evolve a way around it. Whereas if initiating this kind of selective pressure was sporadic, you wouldn't necessarily have the pressure to evolve such a mechanism.
Mm-hmm. Right.
So I guess we've actually been inadvertently addressing Jonathan's question. Are there any comments about survival of the fittest with respect to the virus in light of this? So he's basically, I take it he's asking whether there could be resistance, and you can. It's tough, okay, but you can come up with ideas,
Yeah, viruses always, as Abbie said, viruses always figure it out.
Mm-hmm.
They're really good at this. So I asked a colleague of mine, who I will not name for his opinion, and this is someone I have great respect for. He said, PLOS One is basically a non-reviewed journal. So
Right,
let's explain PLOS One is.
Yeah.
It's not non-reviewed, but the science is just reviewed for the technical aspects, right? Are the experiments done properly, statistically, all that stuff, controls, and then they're published, which I actually like.
Yes.
There's no saying, oh, this isn't significant or it doesn't have high impact. Just publish it and see what happens.
And, a non-scientist might not know there is politics involved in getting things published. And so, I like having an outlet for someone to publish something that's a little different like this that might not necessarily be accepted in a more traditional journal.
Yeah, that's a really good point. You can imagine this going to a more traditional journal and everybody, the editors sort of poo-pooing it and saying, no way. So it is a good outlet for a paper like this.
I'll bet they tried Nature or Cell or something.
Yeah, I'm
and
sure.
They just wanted a billion other experiments, which is always the case. And I think that this is a great journal because of that. I really like it. So I don't have an issue with that. His other point was I'm skeptical about large proteins which have to be introduced into cells. There are 3 problems with such an approach: delivery, and delivery. Yeah, so that's a big issue.
Yeah, that was my first reaction. But as I said, I was not aware of these targeting things, and they present data that it works. Now, down the road, efficiency is going to be an issue, but, and proof of principle is very interesting.
And he said large proteins will induce an unwanted immune response.
I think that's a biggie.
Yeah, probably. Yeah.
You would make an immune response to these proteins.
Yeah.
Now, I was thinking about that as well. Now, I don't know if you had a lot of these domains they're using are proteins that exist in cells. So if you've got human PKR or human double-stranded RNA binding domain and a human death domain of some sort, an anti-apoptosis domain, it could be that those would look like self.
Could be.
Right? So maybe you can get around this. Nevertheless, I think that's a big issue that's going to have to be addressed.
Right. So the person I spoke to didn't think this was prime time, but it may not be prime time, but it's interesting, and I think it should go on. It's definitely worth pursuing.
Absolutely.
Is that our consensus?
Yeah.
And it will be I will be interested to see what will happen to this. It'll be interesting. Could pan out, could fall flat on its face. We'll see.
It's the way it should go. Yeah, you figure it out. Publish it and let the field figure it out. All right, let's do the next one. I think we could do this for a bit.
Time?
I think we have some time, yeah. And this is a paper that Alan had flagged a while ago, and I put it on the agenda because Abbie probably knows about this. This is a Nature Medicine paper called Genetic Impact of Vaccination on Breakthrough HIV-1 Sequences from the STEP trial. Are you familiar with this study, Abbie?
Well, the deal is we have tried many different avenues for an HIV vaccine. And we have ideas that we think will work, and they work with other viruses, and we try it, large scale, 16,000 people in HIV vaccine trials like this. And then 2 years later, we realized that, absolutely no protection is provided to the individuals who got the vaccine, so we have to cut everything short. And then we start back at the drawing board and try to figure out a new approach. So what's interesting about this particular paper is that, okay, yes, people weren't protected by the vaccine, but what happened to the virus in the individuals who did get this vaccine? Because, they've gotten a vaccine to HIV. They should have particular antibodies and cytotoxic T cell responses to HIV. And if they do, then that could direct the evolution of HIV within that particular patient. But if they didn't really elicit a very good response to that particular vaccine, then even though, theoretically you already have antibodies to a particular variant, that variant might evolve in that patient just fine because there was no selective pressure based on the vaccine. So I think that's actually a very interesting paper, because it showed that, even if you don't have the right things in a vaccine, you can direct the way HIV, or you can't direct, you can prevent HIV from evolving in certain directions, because these patients didn't have particular variants of HIV that this vaccine should have blocked. So it kind of cut off evolutionary pathways in patients, and I think that's interesting. So if we knew, maybe particular variants that, individuals progress to AIDS more quickly, maybe we could put that in a vaccine and we could prevent them from ever evolving in patients. So maybe you're infected with HIV, but you don't develop the more aggressive versions. Alternatively, what this tells us is that, there are many, 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 many roads to evolutionary fitness. And an HIV vaccine, in this case, was basically putting up one roadblock. And HIV can't go down that road, but it's got a million other roads it can go down, so it doesn't really care.
Yeah, this was a trial that had failed, right? It didn't provide any protection.
I don't know that we've talked about this trial in any detail, but it was a big trial. We're talking 3,000 individuals in North America, the Caribbean, South America, and Australia. And it was a trial with an adenovirus vector expressing GAG, PAL, and NEF. And in fact, they stopped it partway through because not only was it not doing anything, but they were seeing a higher infection rate in the immunized people. And we've talked about that before. The idea was that the adenovirus, because it was immunogenic, was stimulating production of the cells that HIV likes to infect, and actually creating a problem. But the interesting thing about the paper is, and I should say, because I always like these sort of details, they got virus from 66 volunteers, 40 40 people who had the vaccine, and 28 who were placebos. So they're comparing people who were vaccinated and people who were not, essentially. So they got 66 volunteers. They got a virus from them, but as we've discussed before, any individual generates many different genetic variants of the virus. And so they sequenced many different viruses out of each person. So they had over 400 full-length genomes ultimately that they sequenced, and they can compare the genetic distribution of viruses in people who did or did not get the vaccine.
It's amazing, actually. It's a huge amount of work.
It's a huge amount of work. And one of the things that interested me, it just caught my attention, is that they said they got, they have a way of saying it, I can't quite find it, but they're sequencing, they're not doing bulk sequencing. They're sequencing individual genomes. So they actually dilute out the virus or the virus RNA to the point where when they're amplifying it to do a sequence, they're amplifying from a single genome rather than just doing bulk sequence, which is an important part of the whole thing. They're saying you got a person with a mixture of viruses. If you really want to know what those viruses look like, you have to somehow get the individual viruses.
Yeah. So when they look at all these sequences, this is my understanding, they find some pressure when they compare vaccinated versus non-vaccinated. They find some pressure on a predicted or potential T-cell epitope. That's what they call it.
Right. Mm-hmm.
So does that mean they don't actually know if this is a T-cell epitope? It's just predicted by, software approaches?
Yes, you can actually use computer programs to predict MHC epitopes. Mm-hmm. Of course, you can't do that with antibodies because they're three-dimensional, but with what gets presented in MHC, you can actually do that. At the same time, no one has ever purified MHC molecules and done sequence analysis to see what kind of HIV epitopes are literally in the MHC molecules. You can say that an individual has T cells that respond to a particular epitope, But just because they have those, it doesn't mean that they are actively combating the infection if HIV is never actually presenting those epitopes. So it is a little controversial, but predicting these epitopes is fairly standard in the field, at least.
I think, in fact, they said they tried to look at CTL responses, but in the vaccinees, but they were really weak.
So one of the things, now, correct me if I'm wrong, but one of my impressions was that there was a lot of bioinformatic analysis that they did comparing sequences where they saw no differences. It's as if they didn't see differences until they really zeroed into these very targeted regions, like these T-cell epitopes. And the impression that I got was that although you can see a difference, it's subtle. Is that a fair statement?
Yeah, or they see a difference, but it's restricted.
Right. Okay. So it's not the kind of thing you're going to see just looking at global sequence. You really have to
you have to
zero down into the immunodominant epitopes or something like that.
Yes, and that's going to be different depending on each individual's MHC alleles.
Okay, fine. Okay.
So
the other thing that they saw was that the differences they saw were in the genes that were represented in the vaccine and not in the other genes, okay, which is further evidence that the vaccine is actually driving this.
Yeah.
Yeah, exactly.
But at the same time, all the individuals were infected and all of the individuals, I'm assuming, are on antiretrovirals or at some point will be progressing to HIV. So it's, what? We've had so many negatives in HIV vaccine research world that even finding something as small as this and how we can use this information to inform future vaccine design, I'll take it.
Yeah, absolutely. I mean, the interesting thing is that in a way, the vaccine worked, right?
Yeah.
Because a certain targeted subpopulation of viruses were prevented from proliferating, okay? But it's the standard problem that the virus is so slippery that it can get around that and make viruses that evade that vaccine and cause a problem.
Yeah, it's just like you never got it.
So the vaccine worked. You're saying, Rich, the vaccine worked because it produced a response, a CTL response. That shifted the population in some individuals, right?
Yeah, and that's an important piece of information. Without that information, it's just like, well, the vaccine didn't work, okay?
Yeah.
But it is doing something. You're getting an immune response that's influencing the population of viruses. So that's an important piece of information.
But that immune response did not influence whether there was an effect on infection by the vaccine, right?
No, because ultimately, you make enough virus that can circumvent that immunity, and in terms of clinically, you couldn't distinguish between vaccinated and unvaccinated.
Yeah. So Abbie, if you have this information, they have this one residue. It's GAG amino acid 84. They say it's the most significant signature site distinguishing vaccine from placebo recipients. How could you use that information in another trial, say?
Well, I would like to, I disclaimer, I study more of how to make a vaccine to generate antibodies because I'm not personally convinced that the CTL vaccine is the way to go, just because each individual is going to need a different formulation depending on their MHC type.
Right.
So things get really complicated when you're dealing with CTL vaccines. Yeah. And so the problem then becomes, okay, that's great. These particular individuals with this particular MHC type can be protected from HIV with this particular nucleotide at this particular location. But there are so many different kinds of HIV. It's like, okay, great, you can protect individuals sometimes from this. So you really need to go in and find as many of those locations as you can and try to get them all into one vaccine. And, with all of their powers combined, maybe they can prevent the initial infection. Mm-hmm. And really, that's what you need to do with HIV is prevent the initial infection. It's not the case where, maybe you get exposed to chickenpox, even though you've gotten the chickenpox vaccine, and you kind of fight it off just a little bit and you get rid of it. You can't have those initial rounds of replication with HIV because it diversifies so quickly.
I wonder if any of our vaccines actually do that, prevent infection.
Or more just put the kibosh on things after.
Yeah,
it kind
right.
Yeah.
I think that's probably correct, Rich.
I think that's probably the case. So we're asking for a lot in that case.
I know for polio, it absolutely does not prevent infection. Mm-hmm. You get some infection depending on the vaccine. So I think that may apply to most.
So one of the things they propose for this is, what you can do with this information is to construct vaccines that target regions of the virus that are control regions and can't be changed without, the virus can't change them without becoming debilitated. Mm-hmm. Right now, I don't know if that's a really practical consideration, but that's their idea.
Well, at this point, that's more theoretical because, you guys know you can't force someone to make a particular immune response. Just because there is a theoretical epitope in your vaccine. That doesn't mean that's ever going to be presented, and that doesn't mean that it's ever naturally presented in the virus. Right. HIV has been selected evolutionarily to present certain epitopes, to expose certain things to your immune system and not expose other things. There's a reason why constant regions are constant in a virus that changes, crazy all the time. Right. So to say, we just have to target the constant regions is a touch too cavalier.
Yeah. Okay. Yeah. You're right. It's kind of like I hadn't thought about it before. Is that, the constant regions may very well they can be constant because the immune system doesn't ordinarily see them.
Yeah, that's a good point. Before we leave this paper, I want to mention the first author is Morgane Rowland, and the last is James Mullen, and then there are 30 or 40 in between.
Yeah.
A lot of people on this paper.
There are lots of people on a lot of the HIV papers these days just because, you have to sift through so much data. Yeah. There's no way that smaller labs could do it. You need basically a stable full of grad students to get this stuff done.
Is that the case for your project?
Not at all.
No.
I am the person working on my project.
Okay.
And so things go a little slower, but, I like it. I like being in charge of what I'm doing.
So I presume you're going to publish that at some point, right?
Yes. Working on a couple of publications right now that will hopefully be out in relatively short order. You know how that goes after you submit. And then I've got a few more things in the works. So a paper like we just talked about is more of sifting through a lot of data, and there's a lot of work behind doing that sort of thing. I do HIV research a little differently in that I like to look for problems in HIV research world and try to figure out solutions to those problems. So individuals doing research like, we just had in this paper have an easier time of doing it. So I'm really more about inventing new tools to make studying HIV in the lab easier or more efficient, better. So yeah. That's what I do. I'm more of an inventor.
I'm looking forward to your papers.
They're cool.
So, Abbie, we have reached the point where we move on to emails. If you want to get back to your lab, we can let you out now, or you could stick around. It's up to you.
I think I better get going.
Let me read one short one because I'd like your input. This is from Valerie. She writes, virology fascinates me, but a lot of it goes right over my head. Can you recommend a basic or beginner's book for people outside the medical field so I can start from the ground up? Something that dumbs the complex ideas down for me, like a Dummies For or a Complete Idiot's Guide to book would be ideal. Thanks a lot. So do you know anything that fits that bill?
I think we should get together to write one because I get asked this question all the time. And all I can do is say, go to my blog because I try to write in a way that a normal person could understand what the heck is going on. But at the same time, I'm kind of building up on previous posts. So if you haven't read a couple of my previous ones, I try to link back to them. It might not make as much sense as if you have been following all along. But I don't really know of any good, basic for a normal, sane human that wants to learn more about viruses. I really don't have a good reference for them.
So I know there is one book that I've read which is pretty good. It's by Catherine Crawford, I believe it is. Her name is The World of Viruses. No, that's Carl Zimmer's book. I'll have to put a link in the show notes, but that's the only one I know about. You know of any, Rich?
No, I don't.
Okay.
What I was just thinking is that we need to pick up on Virology 101 again.
Yeah, we did let that lapse by, didn't we?
Yeah, because, if you string all those together and somebody's got the patience to listen to them, I would hope that they're reasonably understandable. But even that is, we don't dumb it down.
No.
Okay.
No. Yeah.
So, you need to write one.
Okay, Abbie, thank you so much for joining us. We appreciate it.
Well, thanks for having me.
Yeah, thanks a lot. It was great having you.
You can find Abbie at scienceblogs.com/ERV. Check it out. And thank you, Abbie, and see you later.
Yay, thanks, guys.
Yep, bye-bye.
See ya.
All right, let's do a few Rich Condit.
Okay.
Let's see, where are we? Okay. Shelby. Ah, yes. Shelby writes, I'd love to hear your thoughts about chicken pox and if it is really beneficial in the long run to get the vaccine. Now, Rich, I presume he means chicken pox, not herpes zoster, right?
Yes, but, they both creep into the conversation.
All right,
because it's related.
You got thoughts on this, Rich?
Well, first of all, since you brought it up, I think we need to make clear what disease we're talking about. Chicken pox, I think most people are familiar with chicken pox. It typically is transmitted as a childhood disease. What kids get it when they're, anywhere from 2 to 4 or 5 years old, and get lesions all over their skin. It's a herpes virus. You catch a respiratory infection, it goes systemic, you get lesions on your skin, and then it establishes latency in your nervous tissue, and it can recur later in life, in particular under conditions of stress or immunosuppression. And then it shows up again as a blistering disease, quite localized either on the head or the trunk, and is known as shingles. And that's apparently a very, I've not experienced it, it's very painful. Okay, and can be a really serious disease. Your kids must have had chickenpox, Vincent, right?
They had chickenpox vaccine.
Oh, is that right?
Absolutely.
I guess your kids are younger. My kids all had chickenpox. It was exciting.
I had chickenpox. In immunocompromised kids, it can be lethal.
Right, and so that's part of the issue is that, when I first saw a chickenpox vaccine, I thought, okay, this is not all that big a deal, okay? My kids had it. It's a rite of passage, that kind of stuff. But you talk to a number of physicians, and they're all for it because they see the cases that get bad. Yeah. And when it's bad, it's awful.
It's horrible.
And you can die from it. It's a really horrible disease.
It's a disseminated disease. These kids look terrible. Their organs are destroyed. Their skin is a mess. I have photographs in clinical books. It is really bad. But let me give you an anecdotal story. So my kids had all chickenpox vaccine. They never got chickenpox. One of their friends lives down the street, comes over one day. He's covered with chickenpox lesions. And I said, well, what happened? And he said, my dad didn't think I needed the vaccine. He'd been home from school for a week. He was miserable because they're painful.
Right.
They can get infected with bacteria, and then you have more problems.
Right.
So he would have been a happier kid in the long run, and he wouldn't have missed a week of school if he'd had the vaccine.
So there's another issue now, okay, now that the vaccine is widespread. It reduces the probability that you're going to, if you're not vaccinated, it reduces the probability that you're going to acquire a natural infection, okay? Which a consequence of that means that it increases the probability that if you do get a natural infection, if you're unvaccinated, it will be later in life. Okay. And if you get infected when you're older, the chances that you're going to get a really serious disease are much, much, much greater. Okay. So, in the context of most of the population now being vaccinated, it is even more important to get vaccinated. Okay. Because you're putting yourself at greater risk. Now, it's an interesting vaccine because this is a live attenuated vaccine, and this is a virus that establishes latency. So the vaccine virus actually establishes latency. And there were questions initially as to how this was going to impact on shingles. Could this thing actually prevent shingles? Would it recur and cause shingles? What's the impact? And it seems like it's okay. It doesn't cause any bad news down the road. Plus, the same vaccine is used in a higher dose, basically as a booster for people who are over 60 years old as a shingles vaccine. And there it has utility as well, because shingles is a really nasty disease. And one of the things that ultimately persuaded me about the shingles vaccine was a conversation I had with a guy who works on varicella zoster, the virus that causes this. And he said, what you really don't want to have is a condition called postherpetic neuralgia, where you get the disease, you get shingles, the disease goes away, it looks like you're cured, but there's some sort of permanent nerve damage, so you feel like you've got shingles indefinitely. Not good. I don't want to do that. So, bottom line, the chicken pox can be a nasty disease, and the vaccine is a good thing.
Okay. There you go, Shelby. Our next one is from Juanjo, who writes, as a plant virologist working in a research institute in Spain, I've been waiting for a good occasion to communicate with you and the rest of co-hosts of TWiV. I started last February to listen to the shows during my daily commute trips to the Autonomous University of Barcelona. I got hooked, mimetically infected, almost instantly, and since then I wait anxiously for every new chapter while hearing backwards in your archive. At a pace of 4 to 5 chapters a week, I'm afraid I'll soon be running short of new materials. Please receive my deepest gratitude for this truly entertaining and educational initiative.
Juanjo writes English very well.
Yeah. Juanjo, you should cut back to 1 a week. I mean, I know you like it. 4 to 5 is a lot.
Yeah.
You want to hear our voices 4 to 5 times a week? Wow.
Nightmares, man.
Motivation for this letter comes after your nice conversation with Professor Raul Andino, TWiV 138. Dr. Andino was extremely didactic when explaining his recent work on RNAi suppression in insect viruses. He was also able to provide a comprehensive revision of the current knowledge on RNAi mechanisms and how viruses deal with this. When commenting on the mode of action of plant virus suppressors of RNA silencing, he was right to say that most plant viruses have adopted a small RNA sequestering strategy. Interestingly, a few exceptions are also known, and some plant viruses target the RISC complex, as the cricket paralysis virus does. In addition to polarivirus P0 and cucumovirus 2b proteins, shortly after the Nyack et al. Paper appeared and demonstrated the activity of CRP1A on Drosophila argonaut 2, 2 independent studies with turnip crinkle virus and sweet potato mild model virus showed an additional mode of action based on antagonizing host argonaut. These 2 unrelated plant viruses have got RNAi suppression activity by mimicking cellular argonaut interacting proteins which contain tryptophan glycine or glycine tryptophan conserved motifs. Indeed, this new strategy adds up an example of the broad variety of modes of action of viral RNAi suppressors. That's really neat.
Yeah, great stuff.
Wow.
2 comments. One, I love the names of plant viruses. Sweet potato mild model virus, and what was this?
Turnip crinkle.
Yeah, turnip crinkle virus. Great stuff. And, we were just talking about the notion that a virus could evolve a way to get around Draco. And these are good examples of viruses evading a really complex immune system by various modes of action, mimicking the RNAi pathway molecules. So
It's very cool
they've
how
got
this
quite an
happens in
imagination.
plants,
Yeah.
and how studying plants can reveal all this basic stuff, which was really the theme of that Andino episode.
Yeah, right.
The final comments of Professor Andino and yourself about the importance, usefulness, and basic interest of keeping up the good work with model organisms like Drosophila, Caenorhabditis, and I should add Arabidopsis in the case of plants were also quite opportune. Thanks again for TWiV, your faithful listener. Yeah, there you go. I was just saying that, and there it was in the last paragraph.
Right. Great.
So Juanjo is at the Center for Research in Agricultural Genomics in Barcelona. Wonderful. Thank you, Juanjo. The next one is from John. Hey, Rich, why don't you read this one?
Okay.
Not for any particular reason, but
Right.
why not?
Why not? Dear Twivers, I have a couple of questions related to TWiV 138. That was the same episode, right?
That's
With
right, yeah.
Raoul, where you discussed short-term adaptive immunity in insects based on RNA interference. As I understood it, double-stranded RNA injected into insects causes a kind of systemic immunity based on getting cells all over the insect to produce siRNA that will mark the viral RNA for destruction inside the cell. Is this more or less right? Yes, I believe so.
Mm-hmm.
If I understood right, your guest said that the immunity to viruses from the siRNA lasted for a week or two, which I guess is a substantial part of the fly's lifespan. Yes, that was correct too. I remember that. The first question this raises in my mind, is it a common consequence of virus infections to have viral genetic material, DNA, RNA depending, end up floating around outside cells? It seems like this would happen when cells burst open with newly made virus particles, and could also happen just from random luck with viral DNA or RNA slipping out of the cell when something else exits. And any kind of immune response that kills infected cells could also leave some leaking DNA or RNA. It seems like it would be true both in insects and in mammals. I also wonder whether, under normal circumstances, there's a lot of host DNA or RNA floating around between cells. This made me wonder how the insect cells could distinguish self-genetic material from non-self genetic material. The mechanism your guest described, if I got it straight, involved double-stranded RNA. Healthy animal cells shouldn't be producing double-stranded RNA, so maybe the answer is that insects only respond to double-stranded RNA. But this made me wonder if he or other researchers have tried single-stranded RNA viruses or any kind of DNA viruses to see if similar immune mechanisms are there. If they do, then I guess there must be some kind of mechanism to keep from having all of the insect cells make siRNA against some protein they would need to survive. Finally, I was curious about how this ties into DNA vaccines in mammals. The fact that DNA vaccines work at all suggests that having viral genetic material floating around must be a useful signal that there's a viral infection that needs to be fought off. But I don't feel like I understand the mechanisms there. It seems like some antigen-presenting cell must be taking up random DNAs it finds by the side of the road and expressing its genes. Is that right? Would the same sort of thing work with viral RNA? I'd love a show where you talked about DNA vaccines. Anyway, thanks again for the wonderful podcasts, which have really opened up a new world for me, and for answering my amateur questions. John. Not amateur questions at all. They're very good questions.
So the DNA vaccines, yes, they work because the DNA that's injected is taken up by an antigen-presenting cell. The DNA is expressed there, and the epitopes are expressed. That's the general idea, right?
Yeah.
I think we should have a show on DNA vaccines. That would be great.
Absolutely.
We should identify someone.
That's a good idea.
Explain the whole thing, because we talk about papers all the time where they're used. Let's see, the question of how the RNA moves around.
Yeah, is there nucleic acid between cells, outside of cells? I've never thought about this.
So I asked Raul, and he said this is an interesting question. They're working on trying to figure out whether it's the virus or the nucleic acid that is spreading within the
Okay.
insect. I don't think that they know the answer.
Okay.
Now, I suppose we have listeners who know this, so you can weigh in, but that was my take from Raoul.
Now, some of the pattern recognition receptors that bind DNA and RNA, are some of those not expressed on the cell surface?
Yeah, some of them are and some of them aren't. Right.
So that implies that there is an advantage to the cell to be able to sense extracellular RNA
Yes.
or DNA.
Yes.
So that implies that this spilling out of the cells or having nucleic acid between the cells is a real deal.
Yeah, I think the idea is that virus-infected cells burst and they release particles and nucleic acids, and these can be sensed from receptors on cell surfaces or by being taken up into cells by endocytic mechanisms. It really depends on the cell that's involved. Now,
also a key signaling molecule, he's correct, is double-stranded RNA.
So double-stranded is key, and as we said in the paper earlier, that's probably very virus-specific, maybe not 100%, but long double-stranded RNAs are.
So that's where the self-non-self thing comes from.
Right.
And even if you're a single-stranded virus, you'll have a double-stranded RNA intermediate in your replication. And even if you're a DNA virus, it's not unlikely that you'll make some double-stranded RNA.
But there's another recognition of RNA, and that's for single-stranded RNA. So some viral single-stranded RNA is recognized as foreign because it has 5' phosphate.
Okay.
And cellular single-stranded RNAs typically do not. At least the ones that are in the cytoplasm. They have a cap, or they have some other. They're in a ribosome and they're not accessible. All right. So the single-stranded RNAs that you would find in the cytoplasm generally don't have 5' phosphate unless they're viral RNA. So there are a number of sensors that can recognize that as foreign.
Yeah, actually, you just reminded me. There's a paper that I don't know if it made it onto our queue that talks about the ribose methylation on the first base of the RNA, and that can be stimulate innate immunity or the absence of it.
That contributes to non-self as well recognition.
Right.
And then there are also probably sensors for DNA. And people are working on this. Some have been suggested, but it's a bit uncertain at this point. But you would guess that yes, so viral DNA has to be recognized as foreign. But how that happens is puzzling because it's double-stranded DNA, and that's what we have. So,
well, if you start off with, I'm making this up, right? But if you start off with DNA on the outside of the cell and it gets taken up and winds up in the cytoplasm, it could be, DNA is not supposed to be in the cytoplasm. Maybe that helps cause a problem.
Yeah, but a lot of these viruses don't go there, right?
Right. I'm talking about taking up DNA that was outside the cell,
just
going back
DNA.
to
Yeah, okay. That could be, yeah. So anyway, we could do another episode on that as well. There are a number of interesting papers on DNA sensing.
So those are good questions.
Okay, let's do one more.
Okay.
Actually, before we do one more, I want to get back to Valerie's email. So she's the one that wants viruses for dummies. So there are two books I suggest. One is called The Invisible Enemy by Dorothy Crawford. It's pretty basic, all right, and it gives you an overview. And the other is Carl Zimmer's new book, which is A Planet of Viruses, I think.
Right.
Have you read that one?
I have read that one. It doesn't, it talks about individual viruses in a sort of an easy way, but it doesn't, when I think about learning about basic virology, I think of a couple of chapters that would be, the fundamentals and that kind of stuff. And Zimmer doesn't really do that.
He doesn't at all.
Jumps right into specific viruses. It's a good read to get interested in specific viruses, but it doesn't really tell you the basics.
I would love to.
Does Crawford's book do that?
Yeah, I think it's very similar to Zimmer, but more extensive, more detail.
Okay.
She is a virologist, so it's better in that sense. But there's no virology for dummies. I would love to write that. I really would.
Yeah, like you've got time, right?
I don't have time, but I want to write it. So if anyone out there knows how I can write a virology for dummies, because that's owned by a certain publishing. And we can all do it together. I don't have a problem with that.
Why not?
All right, the last one is from Cliff. Hey, guys, my name is Cliff, and I'm new to the podcast scene. I started out with a nursing podcast and found my way to yours via iTunes. Now I would like to begin by saying I am not a scientist, like most everyone else that emails you, but I am enjoying learning about viruses. The first time I listened to you all, I actually laughed at my first scientific joke, and then I knew I could make it in the healthcare field. You are educators, and you have become good friends of mine while I work to maintain a woman's building in Ann Arbor. As you thanked Al and TWiV 132 for spending the afternoon with you, I would like to thank you for spending days with me this summer. You guys are like friends of an athlete that never leave his side until he loses his flair. Except for me, it's more like you won't leave my side until my MP3 player dies. I feel like I can be chatty with you. I can even be sarcastic with you. He is having a conversation with us, isn't he? Athlete talk is bringing me to my point. I wanted to email y'all after listening to episode 133, where I think it was Rich asked Kathy if she spoke with Magic Johnson recently since he played ball where she works now, U of M, with HIV. Now, over at MSU, where I participate in the decathlon and I'm working on getting into the nursing program, I noticed a big statue of Magic outside the Breslin. I guess MSU loves him even though he went to U of M. There's the sarcasm. But not a big deal. I'm starting to like all you guys, even with the mistake. I think someone else corrected us.
Yeah,
before.
yeah. So yeah, that's right. We've been over that one.
Also thought I would share a virus reference I like with you. In the first Matrix movie, Mr. Smith speaks of how humans aren't actually mammals, but there is another organism on the planet that are like humans, viruses. You multiply nonstop, use up the resources in an area, and then move to another. You are a virus of this planet.
Okay.
Did you see the Matrix movie?
I saw the Matrix a long time ago, and I haven't seen the whole series, just the first one.
So there is a scene where Mr. Smith is talking to the guy with the bald head.
Okay.
What's
Don't
his
ask
name?
me.
I can't remember. You guys listening are going to think we're idiots. I can't remember his name. He was kind of the boss of the rebels, and he's captured, and he's telling him he's a virus. This whole scene where he's saying, "You're just like a virus." It's a cool scene. That's what he's talking about. Because I'm not a pessimist. I just thought you would enjoy that bit if you haven't seen it. Now, to make this worth your while, I will ask a question. This may not be a good one, but I will ask anyhow. Since sickle cell anemia is a malformation of a blood cell, I learned from y'all that it was in response to malaria and becoming resistant to it. I would guess there may be different receptors on it as well. Now, for some people, sickle cell can be a big problem for them, even if half of their blood cells are normal. So I'm wondering if it would be possible to create a cure using a virus that would specifically attack sickle cells in the bloodstream. But then again, sickle cells would probably continue to be produced by the host, since I'm guessing it's in their DNA to produce them. I could probably Google it to find the answer, but I would rather hear your thoughts on it. After all, you are my friends while at work. I will come back again with some better questions as I start my nursing courses and they arise. Until then, cheers, Cliff.
Crazy. So, sickle cell actually is results from a single base change in the hemoglobin molecule, which causes the hemoglobin to malform, and as a result causes the cells to malform. And I suppose there could be secondary effects on the cells that could influence how viruses interacted with them, but I wouldn't necessarily predict that. It's the mutation is in the hemoglobin that causes the cells to work to sickle. And that actually is, I don't know how that provides resistance to malaria, but it does.
Yeah, they have problems. They have problems getting into those cells. Yeah,
but I wouldn't necessarily anticipate. I mean, it's an interesting idea, but I wouldn't necessarily anticipate that you could find a virus that would specifically target sickle cells, because I don't imagine that they'd have, different surface receptors or anything like that. But maybe we need to ask Dixon this and revisit the malaria thing, because I don't know exactly what confers the malaria.
Yeah, actually, I'm looking it up here. It says it's not really understood how.
Okay, great.
The sickle makes you resistant to malaria, being a heterozygote for the mutation.
And you are right. Cliff, that you still go on making sickle cells because it's a genetic trait.
It's in your
It's in
DNA.
your DNA to produce them.
Now, Cliff, you could in theory correct the problem using a virus by delivering the normal gene, right? By gene therapy.
Right.
So that has actually been looked at, and I'm not sure what the status of that is, but people have. It's an interesting one to try for gene therapy because it's a single mutation. We know what it is, and you can supply the wild type gene. Okay, let us do a few picks, and I would like to go first, Rich, if you don't mind.
Yep, go for it.
I was listening to you and Alan on TWiV 145, and someone said you should do an episode on the history of this recombinant DNA debate. You remember talking about that?
Yep, absolutely.
So I got to talking with Steve Goff, and he said Paul Berg would be a great person because he sort of organized the Silomar conference, and he was a big player, and we could get him. And then I said, wasn't there something going on in Cambridge, Massachusetts? He said, yeah, the mayor decided to block recombinant DNA on his own, research on his own. He said, no more recombinant DNA research in Cambridge because he felt like it. He thought it was dangerous. So I started looking this up, and I found this great video called Hypothetical Risk, Cambridge City Council's Hearings on Recombinant DNA Research from 1976. So this is the mayor, Alfred Vellucci, very colorful fellow.
I'll say.
Talking with a couple of scientists, including Mark Tashney and Maxine Singer, I think.
Yes.
And it's very interesting.
And Ruth Hubbard. And Jonathan King.
Yeah.
With David Baltimore and Wally Gilbert sitting in the background.
So it's great. It's an old black and white film.
Amazing.
He starts out by saying, "Don't use the alphabet. Explain what you're talking about." The mayor says that. It's really good.
It is. I just found that I was riveted to this thing. I just turned it on to have a look. I wasn't necessarily going to watch the whole thing, and I think everybody ought to watch it because of the intensity of that debate. I mean, we're seeing this. It's an interaction between science and the public, and it's science and politics, and it's very instructive because all of the same sort of intensity and lack of communication is there in that. It's the same stuff that's going on now around a bunch of other issues. And I think, importantly, it turns out, listen, everybody, it turns out that recombinant DNA research is not dangerous. Okay? So all of the big fears that people were having proved not to be true. Okay? So every time it didn't have to turn out that way. But you can't just assume that anytime something comes up like this, that the scientists are all being irresponsible and that there's a disaster about to happen. The fact is that the scientific community behaved in a very responsible fashion and regulated themselves, and everything turned out okay.
Right.
But what an amazing video.
So at this time, 1976, the time this hearing occurred, the Asilomar moratorium was in place, I believe.
Yes.
And there was no need for Cambridge to say you can't do any recombinant DNA.
Right.
They were
exactly.
The Asilomar said you can't do certain things that may be dangerous, and he
right.
didn't need to do this. So
right.
Now I asked Steve Goff, what did you guys do at MIT? And he said anybody who wanted a clone went across the river to Boston and did it at Harvard.
Right.
Where there was no ban. And then eventually the ban was lifted.
So actually the initial letter that precipitated this discussion came from somebody who, I was thinking about this after we had the conversation, who was asking, in fact, about research that went on at NIH to test some of the scary scenarios that had been proposed in the recombinant DNA debate. And this didn't really kick in until later, but I started thinking about that. And yeah, I do remember that there were experiments. And in fact, I wrote Lynn Enquist. And I said, Lynn, when were you at NIH? And told him about this conversation. And he said he was at NIH from 72 to 81 and right in the middle of it. He
Yeah.
made some of the
Yeah.
first lambda vectors, helped invent the in vitro packaging systems for lambda cloning, cloned the first mammalian genes in lambda in Phil Leader's lab, cloned the first HSV DNA and the first integrated retrovirus proviral DNA, and had to do it all in BSL-4, or had to do it in BSL-4. He also said, I asked him whether, in my mind, I thought Malcolm Martin did some stuff with SV40 to see if cloned SV40 would, in bacteria, would infect mice or something. I'm not sure I got that right. But he said, yeah, it was Mal Martin, and Lynn says it was Mal Martin, and he had a mobile BSL-4 lab on the NIH campus where he could do this stuff. So, in fact, Lynn or Paul or Maxine Singer would be people that we could get in to talk about the details of that.
Yeah, I've had conversations with Lynn about those days. He has great stories.
I'll say.
He has
great
really,
stories.
really good stories. And if he would like to talk about them, I think we should have, I could have all three of them at different times, because you can't get too much of that stuff.
Right. So it's a really interesting discussion, and that video that you came up with is fascinating.
Yeah, it's pretty cool.
Somewhere between fascinating and really scary.
Scary, yeah. What do you have, Rich?
Ironically, given the listener pick of the week from Jim that we're going to discuss, I came up with a 3D printer. Had you heard of these?
Yeah, they make 3D models, right?
Yeah, I didn't know about this, and
somebody
sent me a link to this thing. So I've got a YouTube link to a little National Geographic bit that describes a 3D printer made by a company called Z Corporation. And what they show in the video is that the way this company does it, you can take some object and you can scan it with some sort of handheld laser scanner, and it creates a digital copy of this thing as in a piece of software, and then you can feed that to this 3D printer, which basically, in effect, builds layer upon layer of plastic to build up some sort of plastic resin to build up a 3D object that represents what you scan. Now, if you look at the video, there's two other videos linked to it on the bottom, because apparently it's more complicated than that. You don't just scan it and feed it in. You have to then go to the image and tinker with it a lot to fill in some of the details. But in principle, that's how it works. And it's astonishing. I can't believe this is really going to go places.
It's cool.
How long has it been around?
I think it's been around a couple of years.
Wow. Yeah. It's amazing. At any rate, the video is really good. I enjoyed it.
Well, we have a listener pick of the week from Jim, our friend from Virginia. May I offer a suggested pick for one of your excellent podcasts from a new publication called Citizen Scientist Quarterly. The pick demonstrates a do-it-yourself DNA extraction process using some homemade equipment, including a Dremel that has been enlisted as a centrifuge and incorporates a part made on a 3D printer. It looks like a very good project for a high school level science class. Professionals viewing the video will especially appreciate such things as licking the fingers while handling samples and retrieving items that have been dropped on the ground. They have a good sense of humor, Jim.
Yeah.
It may also be a good departure point for lab safety awareness. 16 minutes long gives us the link. I'd recommend anyone who sees the video to also Google the maker, Cathal Garvey, because he's an interesting fellow. Okay, gives a link for that. And that can lead one down several more paths of discovery. A lot of Irish are out of work because of the financial recession, and idle hands have more time to invent new products.
So I looked at this. Did you look at this video?
It's
cool. Pretty good. It's pretty good.
There's a lot of this homemade stuff you can do, but it's good to have an example.
I wanted him to go in and pick out the DNA and just pull it out of the tube. So,
that's the best part. Yeah. All that slimy stuff. Yep.
Yeah. All right.
Jim, P.S. with TWiV 133 on HIV and 134 on norovirus were especially interesting because of the diversity of questions and sidebars, as well as the relevancy of the topics. Hey, those were two where we had guests.
Right. Norovirus was Stephanie Karst. HIV was...
the lady from University of Michigan.
Right.
We're bad guys, you know. We don't.
I know.
Let me look it up right here. It was called
Kathy Collins.
Kathy Collins. There you go.
There you go.
Okay, thank you. All right, that'll do it for the post-invincible TWiV.
We're done?
Yeah, we're done. Sorry.
Dang.
You have fun? You have fun?
Yeah.
I thought Abbie was good.
Yeah.
That
was fun.
She was fun. Yeah.
What? She's a ball of fire.
I would say her blog is irreverent, wouldn't you?
Oh yeah.
Yeah, but that's fine. That's what a
fine.
blog is for.
Yep.
It's very different from mine, but she can teach you things because she has a good perspective, and I think it's cool that she wants to do that. You know.
Yeah, it's great.
So check out her blog. She is at scienceblogs.com/ERV. We are, of course, TWiV at twiv.tv. You can find us at iTunes or at the Zune Marketplace or at microbeworld.org/TWiV. Microbe World has an app that you can use to stream episodes to your iPhone or Android device. You can find that over at microbeworld.org/app. Over at twiv.tv, you can download or play episodes, look at our show notes, and picks of the week. If you like TWiV, tell other people. Go on onto iTunes and leave a comment. And that helps us stay up on top of the podcast list so more people will listen. And we love your questions and comments, of course. Can't get enough. TWiV at twiv.tv. We do have a phone line. Not many people seem to like it. It's 908-312-0760. You can leave a message there, and we'll play it so everyone can hear your voice. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Sure enough. Great fun.
Where there is no hurricane at the moment, right?
No, skipped us. You guys had better duck.
Well,
it's not looking good.
Yeah, that's what I hear. But, you know, up here in the Northeast, we never really get hammered.
Yeah.
It's pretty rare. But we had to batten down our beach house last night. We took our sailboat home.
All right.
Which is too bad because I wanted to sail it some more this year.
I'm going sailing tomorrow. Not tomorrow, Sunday.
Have fun.
Sunday. Yeah. Well, good luck with that storm because if it does hit you, that's not fun.
Yeah, I don't want to have bad damage, but a lot of people down south are probably going to get hit harder than we are, so good luck to them as well. I'm Vincent Racaniello, of course, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.